Okay, welcome everyone to the Rutgers NJAES Ornamental IPM Program webinar for today, April 22nd, 2025. I'm Bill Errickson. I'm the agriculture agent from Monmouth County. I'm joined with our IPM program associates, Steve Rettke and Tim Waller, agriculture agent from Cumberland County, and our support staff, Erin, Quinn. And we're going to talk today about some of the common things that we're seeing in our nurseries throughout the state. So again, disclaimer on the bottom, we're going to be talking about some materials that are for licensed professional applicators, and you always want to make sure that you're following the label, using proper PPE, and you can always reach out to us if you have any questions about anything on materials. Always feel free to give us a call. We can help you out with that. Okay, so just some updates from last time. 2 weeks ago, mites were part of our talk, some of our cool season mites, and we had a discussion and some additional research that we looked into on some miticide materials, and just wanted to give you an update on some of the most effective materials. And the first 6 on that list are all reduced risk materials. So if we're looking for some good miticides, we've got some more options there if you're looking for controls. Another update you'll see at the top of the screen are some projected growing degree days going out for the next few weeks. So our next webinar, our next official webinar, is not going to be for another 3 weeks. So we wanted to give a little bit of a forecast here to give you a sense of what to look out for, what ranges for some pests you might see during that time period. Things are going to be warming up fast potentially. But again, it's just like predicting the weather. This can change. And the farther we try to predict in the future, the less reliable that is. So find a system to check growing degree days. This is from US Pest. Reach out to us if you want help setting that up for your farm. And on the bottom, you'll see, significant portions of New Jersey are still in some sort of drought status. Monmouth County is looking pretty good, but in the north and south parts of the state, we still have some drought conditions. It's encouraging though, we do have some replenishment precipitation coming in. So we're at the normal or above, slightly above normal range for precipitation for replenishment. So we are rebuilding that resource, the water, but we're still in drought in some areas of the state. So just keep that in mind as you're starting to get to work. Another update on our boxwood blight risk model. This is also from US Pest, and you can scan that QR code either now or when we send out the PDF of the slideshow, and that can help get you set up for monitoring specific for your operation. And so we see low risk for the next week in most areas, a little bit of risk in South Jersey for susceptible varieties of boxwoods. But after this week, things can start to progress quickly, and there may be higher degrees of risk for boxwood blight. So we'll try to send out updates through the Plant and Pest Advisory and through our email lists. But because we're not going to have another webinar for a couple weeks, there could be some things that change coming up. So reach out to us or sign up, get yourself set up on one of those models for the most accurate information. Another update we have is on ambrosia beetles. So we talked about ambrosia beetles in some detail in our last webinar, and we're starting to see some activity. Tim has been getting some calls in South Jersey where several farms are reporting adult ambrosia beetles being caught in their ethanol traps. So we've got some growers who have their traps set up, and ambrosia beetles are attracted to ethanol, and they've been finding some adults. So it's starting to be active in South Jersey, and that means it's coming through the rest of the state. One of the key triggers that we look for ambrosia beetles is 2 or 3 days above 70 degrees. And if you get that, that's when the adults really start their flights. And so we see that definitely happening this week in South Jersey. So if it hasn't happened yet on your farm, it's probably going to happen soon. Central Jersey, we may see it depending on how things go. North Jersey, you may have a little bit more time, but it's coming soon. And so one of the things we can do to prevent the damage here is to reduce plant stress. Ambrosia beetles are attracted to stressed trees. And that includes, if you're digging trees right now, that's a stress on the tree. And especially if they're waiting around or they're before they're being shipped, that's considerable time that they could be stressed and they could be getting attacked by the adults even when they're loaded on the truck waiting to go. So another stress can be flooded soils. So when we start to find adults in the traps, some of our contact materials on the lower stems and the lower bark, that's where we want to put that to address the adults. So this is happening now or right in the time of that. That's our ambrosia beetle update. Okay, so some of the new things we're going to be covering today, we're going to talk about the boxwood leaf miner and holly leaf miner, eastern tent caterpillars, some evergreen lace bugs, clear wing borers, spongy moth, the red-headed flea beetle, fire blight, and some rust diseases. So let's get into it. And as we go through, you'll see at the bottom of the screen, our current growing degree days. North Jersey is around 60, Central Jersey is around 120, and Southern New Jersey is 165. So this is going to let us know roughly the range for right now for some of the insects that we're talking about. And the first one I want to mention is boxwood leaf miner. We're a little bit before the adult emergence, but this is a time that we really want to be monitoring and looking for areas where these might be causing problems. So these are going to overwinter in the leaves as larvae, and that's what we're looking at in the picture right now, are larvae feeding inside the leaf of boxwoods. And at this stage of the game, the larvae are actually pupating and turning into adults. So they kind of feed during the spring and then they go into the pupal stage before they turn into adults and emerge. The adults are probably going to emerge within the next 2 to 3 weeks, roughly. So it's a good time to go out and start looking for where they may have laid their eggs and where this damage might be. And this is what it will look like. So you can see like the blistering on the leaves. Folks who grow boxwoods are probably really familiar with this, but the top picture is showing the blistering of the leaves. And on the right is after the adults emerge and they kind of bust out of there. But this is inside the leaf is where the feeding damage has occurred from the larvae, and it can be hard to control them in there. But at the bottom picture is really interesting. So those are the adults, and that's where we're trying to target their emergence for control. They're actually caught in spider webs on this picture. It's a natural population of spiders, and then the adults got caught in the web. So this is what your sticky traps can do. Yellow sticky cards, and we have them available if you need them. Let us know. And this is a distinct looking insect, that orange body on there. So when the adults come out, that's the window we want to target for control. And it's a pretty short window. The adults are only active for like 2 to 3 weeks. There are some natural predators, but monitoring the adults with the sticky cards is key so we can time that properly. We want to minimize plant stress. And if you're choosing new boxwood varieties, there are some that are reported to be more resistant to boxwood leaf miner. You may have to prune out some of the damaged leaves, and you want to destroy that material so they don't hatch out again. So you can see our contact materials. This is what we would want to get down when we observe the adults. So the adults come in contact with those and don't survive. And then systemics can be effective when the larvae start to feed. So the adults are going to come out in a few weeks. They're going to lay their eggs, and then the larvae are going to hatch inside the eggs, inside the leaves, and it can be harder to control once they're inside there. Another leaf miner that we're dealing with now, we may even be seeing some in South Jersey already, is the holly leaf miner. These are also going to overwinter as larvae inside the leaves, and they're going to mine through the leaf again. I'll show you some pictures of this also. And then the adults emerge, and they look like these long black flies. So we've got a picture of a close-up of the adult there, and that can be useful because when we have the sticky cards out, there can be a lot of different insects on there, and this will help to really identify if that is a holly leaf miner or not. So these can be a problem on American hollies and some of the cultivars as well. But this can cause this kind of damage in the holly leaves. And this is a good time of the year to go out and to look for this feeding damage from the larvae. You'll see the mines that have formed there. So the larvae are going to cause that damage, and then the adults are going to come out. You also may see those little tiny holes. So in the picture on the top left and the middle left, the tiny holes on the leaf, those are actually either feeding or laying eggs from the female. So she has kind of probed into the leaf there. And that's something else that you can look for in your scouting and your monitoring, because that leaf may have eggs laid into it. So look for these damaged leaves. You can remove them and prune them out if you have small populations, or if it's, a small amount of hollies. But this can be a good way and something you can do now in terms of monitoring before the adults hatch out and lay the next generation. So in natural stands of American holly, this insect is really not much of a problem. It will cause some damage, but there's a lot of natural predators that take care of this in the wild. But then when we get into a nursery situation, things are always a little bit different. So avoiding stress, again, keeping plants healthy, removing those mined leaves this time of year before the adults hatch out. And then also, if any leaves have dropped to the ground prematurely or for last year, like good sanitation, cleaning that up, because those could still have viable insects in them that could hatch out. Our sticky cards are going to monitor for these adults again, and that's going to help us with our timing to know that the adults are around, and then the contact materials are going to help to control the adults when they land to feed or lay their eggs. Systemics can be used later in the season, but this is only for larva control. So once the adults lay their eggs, the larvae hatch out and start to feed inside the leaves. This is where the systemics can be effective, but this is going to be later June in the season. So it's understanding that insect life cycle that we're looking at for that window of adults that are coming out around now. Okay, so another thing you may be seeing in South Jersey already are eastern tent caterpillars, maybe central New Jersey possible, North Jersey coming soon. And these are going to overwinter as eggs, and I'll show you a picture of the egg masses on the branches. One thing that we can do in the spring is to prune those out if you can find them. Then the yellow and black caterpillars hatch out. They're usually protected by webbing, and they're going to feed on buds and leaves of the trees. They're most problematic on our rosaceous trees, cherries, flowering cherries, crab apples, things like that, but they can also affect other species as well. So if you grow a lot of flowering cherries or crab apples, look at those being like the main trees where you might see them. Bottom left, we see the egg mass there. So if you can find those and remove them or prune them out before they start to hatch out and cause a problem, it'll take a little bit of monitoring and scouting and looking for them. But if you can see them, then you can get them before they cause damage. Up top, of course, we see the caterpillars in the webbing there. And on the bottom picture, the bottom center is really interesting. There's two wasps in that picture. And then you see some slits cut in the front of that webbing. And those are holes that the wasps made to go in there and feed on the caterpillars. So there are parasitic or predatory insects that will help to take care of them. So those can be beneficial as well. So there's lots of natural predators. Birds will eat these insects. Pruning out the egg masses, removing those silk tents, sometimes opening up the silk tents, knocking them on the ground. Birds will eat them. That can be a good way to deal with that. You may be tempted to use fire for control. Resist that temptation and do not use fire, because that can cause damage to the tree as well. It will kill the insects, but will also cause damage to the tree. So we've got better controls. Physical removal is going to be our safest option for non-target organisms. But we can also use contact materials. But we have to get into that web, remember, and a lot of any materials that we're using that are going to be contact or biorational materials that need to come in contact with the insect are not going to be effective if they're protected by the web. A good material we can use is BT, Bacillus thuringiensis, but they have to consume that. So you have to put it somewhere that the caterpillars are actually going to feed. So on the buds or the leaves, those are the parts of the tree that they're eating. And they have to get out of the tent, and it has to come in contact with that. All right, Steve, I'm going to turn it over to you now.
Okay, very good. Thank you, Bill. And good afternoon, everyone. Glad to see some of you back again for this lunch pest hour that we're doing. And so I'll be assigned to speak on evergreen lacebugs. There's a few different species that are becoming active soon if they aren't already active. We have some clearwing moths we'll discuss. There's a few of those also. And then if I have time, I'll speak a little bit on the spongy moth, which is one that all of you are familiar with, but maybe haven't really seen much in more recent years. All right, so the first ones will be the evergreen lace bugs, and I'm going to concentrate on the azalea lace bug and the Andromeda lace bug, but it could also apply to the rhododendron lace bug. They all come out about the same time in central New Jersey. They're hatching now. These are lace bugs that will feed only on evergreen hosts. As the name actually applies, they're quite host specific to the name that they're given. And therefore, when they start hatching, which is now in central New Jersey, they've been hatching for a while down in South Jersey, and they'll start within probably just another week or two up in North Jersey. And so you can see the damage that they do. They have a stippling type injury where they feed within the cells of the plants. And we'll go ahead to the next slide. And this shows a closer look of these symptoms. They're a type of a stippling damage that they do. And sometimes they can be quite extensive and they change the color of the leaves. As these lace bugs are feeding, they are removing the green chlorophyll from the cells. They're killing the cells. And once those cells are killed, the stippling will remain for the rest of the life of that particular leaf. And so therefore, there's no way that the plant, of course, can repair the damage that it has. So, and just to emphasize the growing degree days there, there's actually 2 and maybe even 3 generations of these lace bug species. And they're beginning, as I said, occurring about right now. And they have actually up to 5 nymphal stages. Each of the species have 5 nymphal stages. And so and just showing you there kind of a line drawing schematic of actually that's not a lace bug, because if it was, it would be feeding on the lower leaf surface. So that's some kind of a plant bug there. But you can see what's happening is that with a lace bug, they would move through the underneath, through the spongy mesophyll, and then they begin to feed in the palisade parenchyma cell, which is right near the surface where all the sunlight is. And so they're concentrate their chlorophyll in those locations, and then the lace bug uses its piercing, sucking mouth parts and removes the chlorophyll. And that's a close-up, an extreme close-up actually, of an azalea leaf on the undersides, and that's the primary leaf vein. What this is indicating is the fact that the lace bugs will lay their eggs, azalea, rhododendron, andromeda, all the same, right up against the edge of the primary and secondary leaf veins. And they insert about 90% of the eggs inside the plant tissue. So just a very small amount of it is exposed. And then what the female does is she releases her fecal material on top of the eggs to protect it even further. And so that's why I say there in the upper right corner that oils will not control eggs, because the eggs are not going to be contacted by the oils whatsoever. So, and it's kind of also a little bit curious is the fact that all the evergreen lacebug hosts, the rhododendron, azalea, andromeda, they're in the genus Stephanitis, and they have oval-shaped wings, and they overwinter as eggs. Now we also have a number of deciduous lacebug species, which aren't going to be active quite yet because the leaves really haven't formed to much of a degree at this point in the season. But they curiously have rectangular shaped wings, where they're more of a sharp angle on the corners of each of the wings as opposed to the rounded edges. And they're in the Corythuca genus. The deciduous lacebugs are the Corythuca genus. And that would be things like hawthorn lacebug, oak lacebug, sycamore lacebug. And we'll talk about those probably later in the growing season when they become active. And this showing, just make sure you realize they don't come out as adults, they come out as nymphs, and we only have 3 adults on this particular image, and this happens to be an oak lacebug image, and so those wings have squared off edges, they're more of a rectangular shape, but all those other ones are various instars, and I said that there's 5 different instar stages with lacebugs. And so they keep growing as they get larger. Only when they have reached the adult stage do they have wings, and they can fly. They're not particularly strong flyers, but they actually don't prefer to fly, and you can try to force them, encourage them to fly, and they will with enough force. This, by the way, we'll stay here. This indicates just more of a distant photograph. In this case, it's not, it's a hawthorn lacebug again, but you can see where they're laying the eggs right up against the veins, and then their fecal material is covered up on the eggs, so oils will not work. All right, so there's a lot of good information here. I've already probably talked about much of it already. You know, actually toward the bottom there, it says damage can be more severe in sunny sites. And the reason for that is predators. Predators will be less likely to be found in sunny sites. So many predators prefer more cooler, shaded locations. And so you have better predation and fewer insect like lacebugs in those sites. And finally, as far as controls, don't have time to really go much into detail of this, but the thing is to remember that these lace bugs are on the undersides of the foliage. And so if you're going to be using contact, you have to apply your treatments under the leaves, which can be kind of difficult, you know. And so things like carbaryl or the pyrethroids, they need to have direct contact. The same thing with oils if you're going to go with that. Now there is material, it's an acephate, it's a translaminator, so all you have to do is apply over the top. Another one that works as a translaminator or even as a systemic are the abamectins or the abamectin, and that will be absorbed into the foliage, and as the lace bugs feed, they'll take up the toxins. And then to finish up, the neonicotinoids, the imidacloprid, dinotefuran, they, as soil injections particularly, will give you at least a year, and something like imidacloprid will probably last 2 or possibly 3 years. And so you don't have to do continual retreating. So, right. All right, let's go to clearwing moths, and there's 2 of them now that are becoming active, at least in central Jersey, and that's the lilac clearwing moth and then the dogwood clearwing moth. Those are the first ones to appear, and there's about 6 or 7 different clearwing moths. Now, these are caterpillar borers. You know, most of our borers are going to either be caterpillars or beetles. And so the clearwing moths are caterpillars. And so this shows you the adults of what they look like. The lilac borer on the upper left, you can see that the wings are clear, but they have more of an opaque appearance to them. And then the dogwood borer is really clear wings. You can see why they got the name of clearwing moths. And so, and they can create a lot of damage. Now, this happens to be a green ash tree. You might assume immediately, well, it was killed by the emerald ash borers. And no, it wasn't. This was actually killed by clearwing moths. It was the banded ash clearwing moth, which is kind of a unique one because it doesn't come out as adults until September. So that's the exception to the rule, whereas all the other clearwings are going to be coming out in April, May, June, and July, depending upon the species. And they all often come in an orderly sequence as you go through the calendar. But the dogwood borer and the lilac borer are the first 2 that are active now in many parts of the state. And there it is a caterpillar. And as I said, it's either going to be most borers are high percentage. Beetles or caterpillars are going to be the borers. And with caterpillars, if you're not sure if it's a beetle or a caterpillar as a larval stage, count the prolegs to see if they have prolegs. And if you can tell, I think you can see my cursor. You got 1, 2, 3, 4, 5 prolegs, and they come in pairs. And underneath, there's these stiff hairs. And if you see those, that you know it's a caterpillar and not a beetle, because beetles do not have prolegs. Okay. And this showing you the pupation skin, and they all have these papery thin type pupation skins as they emerge out of the tree after they finish at the larval stage, and then they'll emerge as adults out of the pupation stage. That is not something that's going to last very long. It's very ephemeral, and it'll break apart within just a matter of a day or 2. And I was just lucky I got that particular photograph. And they leave about a 1/4 inch circular hole in them. And so that's very distinctive. And I keep saying some of these traps are available. I know we've already mentioned traps, and these are all, you know, easily available traps that can be used. And this showing you here, again, the growing degree days for those 2 adults and their emergence. And this landscaper is looking at a wing trap, which contains a pheromone, a synthetic pheromone of the females, and the males are attracted to the female pheromones, which is basically a sex perfume, and they fly toward the trap, and then the males get caught. And then on the right, you can see some of the adult clear wings. These are lilac, the ones that are dark gray. They're a little more than an inch in size or so. And then the ones that kind of look like yellow jackets, that black and yellow banding, those are oak borer clear wing moths. Not a real particular problem in the landscape, but they do cause some damage. So. And that's a close-up of one of the clearwing moths. That happens to be a peachtree borer. But and one thing I have to mention is that these clearwing moths, in most cases, most of the species, before the female will be able to successfully lay eggs, there's got to be some kind of a wound on the trunk of the trees. It's either the main stem or maybe the lower scaffold branches. And if they got wounds on them, such as good example, if you got lawnmower blight taking place, wounding the base of say a prunus tree, you're really inviting these clearwing moths to come in and lay their eggs on those wounds. Okay, they do mimic as an adult. They're moths, right? They're caterpillars, but as these clearwing moths, they're day flyers, and they want to avoid predation. So evolutionary strategy is they mimic the appearance of wasps. Also, their action appears to be that of a wasp. And so therefore, the predators kind of leave them alone. I'm going to deal with a wasp. And so it does help them to reduce predation. And this is showing you what you can do with your pheromone traps. You can actually put the pheromone in the tree that you plan to potentially protect. Say that's a dogwood tree there, that drawing there on the left side. You can put the pheromone trap right in the tree. And then as the wind blows the downward plume of the pheromone, you have these adult moths that are now active, and the males will kind of maybe blunder into this plume. They may not necessarily be, they're kind of searching for this pheromone. They have a rather elaborate antennae that allows them to do that. They could directly fly into it, or sometimes they may become at an oblique angle. And so they'll fly through the plume, and then they'll go out of it, and then they'll turn, and they'll go back into the plume again, and they go out of it, and they'll turn again. So you might have this zigzag type of pattern. That's kind of a hypothesized motion that maybe some of these male moths are doing. And so again, some good information here. They're a little more than an inch in size. As far as the lilac clearwing moth, the dogwood borer clearwing is really quite small. It's maybe just about a ½ inch, slightly more. And so that's one of the smaller of the adult the dogwood is one of the smaller ones. Rhododendron is about the same size. And so, but there are ways to tell the appearance of these adults, and I don't have time to go over that now, but oftentimes you can count these yellow bands that are around the insects as shown there with the dogwood borer. There's 2 yellow bands. The rhododendron borer looks a lot like it, but that has 3 yellow bands. So you can use these traps, and you learn how to, they help you with it when you purchase these pheromones. They help you with ID and what to look for. And so just to finish up, with this information, the lilac borer at the bottom, dogwood borer, even though their name indicates they feed on lilac or dogwood, which is true, but they also feed on some of these other hosts as well. So don't just assume that they're only feeding on the name that they're given. All right, finally, with controls, pyrethroids are probably the workhorse because you're trying to time when the females are going to be actively laying eggs. You want to put this protective insecticide on the branches of the trunks, the stems, and you want to time that before the eggs are laid. And so that's why you catch the males first. The males will be active first, the females follow, and then you know that the females, after mating, can then begin to lay eggs. And so if they do lay eggs on the tree you're trying to protect, the egg will hatch, it'll burrow into through that protective chemical barrier, and the young larvae will be killed, all right? And so there are some systemics that are labeled for it, but I think research has shown that they're not particularly effective. Something like emamectin benzoate, which is a great control against things like emerald ash borer, but they maybe just not quite effective against these caterpillars. All right, so this spongy moth, right? Okay, see, I'm getting out of time here, so I'm just going to go real quick and just remember that it's one that has a long history in this country. It kind of started around 1870. Just to show you these photographs, and that upper left photograph indicates a wood area that is completely defoliated, and that can occur with high populations of the spongy moth caterpillar. The bottom left shows a female adult, a white colored, cannot fly, but she's laying her egg mass, maybe an inch, inch and a half in size, has a yellow coloration to it, maybe 600 egg masses is an average size. The one on the further to the left is a male moth. See the extensive pheromone detection with their antennae. And that's kind of a drab moth. It does fly and has a zigzag motion when it flies. And that's what they look like around 5th instar. They can have up to 6 instars. I think the males have 6 instars, rather the females have 6 instars, the male have 5. And when they get to that size on oak trees, and they feed nonstop the last few days before they pupate, and they can feed on 1 square foot of foliage per caterpillar per day. And so they can consume a lot of foliage. And then here's their hatching out. And so we are at 120 growing degree days in central New Jersey. And so they have begun to hatch. As it hatches around 90, and they can, as caterpillars, you got that window for control up to about maybe early June before they really start doing some really heavy damage. But they don't emerge from the top of the egg mass. They emerge from the bottom. Okay, and those are young caterpillars that are just coming out. And this showing you the spread over the last 150 years. They started in Massachusetts. They came to New Jersey about 1960s. They reached their peak populations in New Jersey in the early 1980s. But something happened that caused them to be much less of a problem as the years have gone by since then. Okay, and this showing a good graph of the New Jersey Department of Agriculture showing how they really peaked. And this is the New Jersey defoliated acreage, and they peaked in the early 80s. You can see how high the numbers were. But again, as you move through the years, there was something that happened. Look at that. They've been much fewer in number, with a few exceptions here and there. Like in 2006, 2007, 2008, they kind of resurged a little bit. And what was the cause? Why did they drop so dramatically? And and that's because of this. Back in the they actually had tried to do this earlier in the 20th century. It didn't work well. A bad timing with the release. But they went over to Asia. They found this Asian fungus called Entomophaga maiga. They knew that it was causing suppression of the Asian gypsy moth, and so they tried to release it in northeastern New Jersey, Massachusetts, New York, Virginia, and boy, did it spread. It spreads really well with cool, wet springs, and it's exceptionally virulent against the gypsy moths or the spongy moths, okay? And so that shows what happens is they just dry up. It's a dry fungal rot. You put a knife blade against that, it just crumbles. And I have to really kind of finish up here. I've probably talked about much of this. Just remember, the trees themselves are not directly killed by the spongy moth, but will happen after really severe defoliation, say for 2 or 3 consecutive years of 75% defoliation, it weakens the trees, and this is especially with oak trees, which are their favorites, and then it sets them up for other more lethal type of pests like the 2-line chestnut borer or the armorelia fungal root rot, okay, armillaria, okay. And there's more than 300 species that this will attack. And so finally, just controls. There's a lot of things that control caterpillars. Caterpillars are not difficult insects to control. You have all these contacts, these pyrethroids, carbamates, acephate, they all work well. BT, I'll finish up with this. The one that's kind of an IPM material, the BTK, which is the Karstocki strain, that is a stomach poison. And go to the last slide. There should be one more slide. There it is. These are what you're doing when you're applying BT. You're not applying live bacteria. You're applying spores and crystals. And the bacteria produces these endotoxin protein crystals, as shown there with that red line. And the caterpillars must consume these crystals. And when they do so, it causes their gut lining to be pierced and like little holes being punched in it. And so it's a stomach poison. It causes bacterial septicemia. It doesn't kill the caterpillars immediately, but within less than 24 hours, they stop feeding. And within usually about 3 days, they die. And that's a scanning electron micrograph showing you those crystals. Okay. All right, I'm already over, so let me hand it over to Tim Waller. I think Mr. Waller's up next, or Dr. Waller, excuse me.
Hey, everyone. So I'm going to kick off this section here talking about red-headed flea beetle. This is a problem that we all run into, particularly in container settings. We all know that's got a really large host range. It can be quite difficult to actually find the beetle sometimes, but a lot of times we will see the damages as being evident. And the point I wanted to really start off with before we really start getting into this is you need to make sure that you're targeting the right pest or disease. When we're looking at these plants, I've run into a number of instances where folks were telling me they had red-headed flea beetle, they were treating heavily with insecticides, and they were still having damages. I've seen that in a number of instances. So what I went through is I collected leaf samples, and if we look at those bottom images, we can see those darker regions, that they do look like they have holes in them that could look like they were caused by a chewing insect. However, after running some incubation on those, I was able to determine we're actually dealing with a foliar fungal issue, in this case, Calototrichum, compared to the damages that we often see caused by red-headed flea beetle. So we are going to see adults coming out in roughly a month from now, and that's why I wanted to talk about it at this point in time. So we're rapidly approaching when red-headed flea beetle is going to start hatching. It may already be hatching in some instances. And as these grubs grow, the idea or the plants are going to begin to grow. They're getting much larger. And by the time the adults are coming out, we could potentially be dealing with huge populations of them. In our area, we typically have 2, maybe 3 generations, but as those generations progress, monitoring them with growing degree days gets more and more difficult as time goes on. So we know that there are a number of crops that red-headed flea beetle favors. Itea, Weigelia, and hydrangea are very high on that list. I've also seen it in a lot of different herbs, see it a lot in salvia. It's got a pretty large host range, but it definitely does have some favored plants. And like I was saying before too, red-headed flea beetle is oftentimes difficult to find the adults. And if we are relying our IPM program on finding the adults, then going through with a spray program, I think we could potentially be missing a key first step in this. On the natural predator side of things, red-headed flea beetle is not really that big of a pest once these plants make it to the landscape. They get taken out by natural predators, and you'll oftentimes see those damages rapidly decline once the plants are planted into the ground. Some of the really important cultural practices too are if we have plants that we're holding over from the year prior, it's not a great idea to put our brand new starts right next to the plants that we've been holding over. And maybe we held them over because they had damages the year before, and we had to trim them and push a new period of growth for them. So if we can do physical separation of hosts that we know, or plants that we know that are highly susceptible, and maybe carrying over a population of this beetle, all the better. What I'm really going to stress today, though, is we're getting into egg hatch. So there are larval treatments. If we go over to the biorational side, the azodirect do have some good activity against the larval treatments. If you are in a neonic limited situation, this is definitely an option or could be built into your IPM program. So when it comes to treating at this point in time, there are no adults, so our contact materials, which work well at taking out the adult beetles, are not going to be effective. However, if we were to initiate systemic applications, there's been a lot of research done with various forms of imidacloprid, our neonics. We are preparing those plants such that when the adults come up, they're feeding on a plant that is already toxified by the neonic, and we're going to get a really big hit to that first wave of the population. As the adult populations increase, we'll start doing an update on red-headed flea beetle as we progress with this, and there are contact materials that are very effective. However, they get us away from that concept of an IPM program because they are going to be taking out other organisms that are out there. So what I wanted to kind of drive home here is if we're looking at our grown degree days, this is the south, central, and northern parts of the state. We're starting to have egg hatch, and we're approaching that first generation of larvae, which means in 2.5, 3, 4 weeks from now, we're going to have adult red-headed flea beetles feeding on our crops. Unfortunately, when the first generation comes up, this is often associated with the bloom period, and it's often when you may be pulling these plants to move them out for sale. If you can utilize a systemic material this time of year, it's going to prime those plants so that we're going to have many less adults to deal with as time progresses. All right, so next on our list here, pertinent, time-sensitive kind of thing is a number of rust diseases, particularly looking at the gymnosporangium rust. This is cedar hawthorn rust, cedar quince rust, and cedar apple rust. What we're looking at right here are symptoms that we would, and signs for that matter, that we are going to see in a considerable amount of time from now, maybe a month or so, where we're starting to really see some of these symptoms on our rosaceous hosts. If you do have cedars in the area and you look through them, you may see some of the structures that are over on the right side of the screen here. Those are our artelia. And these are the spores that are coming from the rust primary host, that cedar. So basically with this whole group of rusts, we are dealing with two host species. We've got our juniperous hosts, our eastern red cedars, and we've got all of the different rosaceous hosts that we grow in our nurseries. Management in the cedars is something that can be done, but often is not, because these rust spores can travel great distances, 2 miles or more in air currents. So that may not be a reasonable approach. However, we can protect our plants in early spring by targeting our rosaceous plants. Just some background on them. When we're seeing these bright orange colorations or these rusty, or we're starting to see these abnormal growths growing out of the bottoms of leaf surfaces, distorted tissues, swollen tissues, there's a really good chance that we are looking at a rust pathogen. So when it comes to cultural practices, this, like every other disease, if we can increase our sanitation practices, if we're removing diseased materials, we're getting infected inoculum out of the system, this can be quite impactful. Avoiding overhead irrigation, increasing airflow, these are all things that are important for all of the above ground diseases that we're going to be dealing with in the nursery. So a lot of the spray program is centered around the DMI fungicides, that group 3 material. There are a lot of great options there. However, that's just one chemical class, and we all know that rotation of our chemicals is really important. So there are protectant fungicides that we can use as important rotational partners. Chlorothalonil is an option, mancozeb or maneb, or and similar compounds can be great rotational partners in this. Just a consideration, many of our malice or our apples, crab apples, do have can have some level of phytotoxicity to some group 11 materials, particularly azoxystrobin. So it's important, if that's part of your regime on these plants, that you're making sure that you're checking the labels. And many of the times these labels will give an indication of what varieties are more resilient to that type of material, because that gives us another option. There are many biorationals that are available depending on what situation that you are utilizing and who the buyers are from your plant material. All right, so this is another one that I wanted to talk about this time of year. So with fire blight, we're not going to see a lot of the damages at this time of the year. We're going to start to really see this as it really heats up. We're getting much more mature leaf tissues. However, during this time of year is when a lot of these initial infections can be occurring. It is also a time, if you weren't able to get in and prune out materials beforehand, you may start to see damages, albeit without the obvious flagging and destruction of leaf tissue. So it's caused by a bacteria. Being that it's caused by a bacteria, that tells us that this pathogen is difficult to control, and we have very limited resources available for it. It thrives during warm, wet weather during the springtime. This week is going to be quite warm, and as we move towards the weekend period, we are going to be getting more precipitation, at least in the southern part of the state. So that's kind of that 1-2 punch of environmental factors that could lead to more spread of this disease. Oftentimes, this will come in through wounds. It can be vectored around by insects, particularly later in the season. And it's notorious for attacking floral tissues, and they're making its way in through the nectaries of those flowers. So it really hits on our rosaceous family pretty hard. And like I said, later in the season, we will start to see those shepherd's crooks. Something that we're going to bring up as we get towards those warmer months, I want to do a couple slides really looking at pruning practices. I know it might seem obvious, but I do get a lot of calls about how far away should I be pruning out these disease materials. Generally, we want to be pruning this anytime that we're trying to prune out a disease. We want that plant tissue to be dry. Preferably, this is going to be a little bit later in the season. If we know an issue is there, we can prune it out when it's not oozing and actively moving millions and millions of bacterial cells around. So we're going to go down 6 to 8 inches is what you see in a lot of literature. If you can go even a little bit farther away from that, better off you'll be. And sanitizing your tools between cuts is critically important within a specific plant, and when you move out to different blocks, sanitation is really important. So a lot of our program relies on streptomycin, but we also do have phosphonates in the aluminum tris group that can be effective, as well as coppers. Just with the caveat too, that if you look at a lot of literature, it says if you're using the phosphonate group of materials within a 14-day period of coppers, you can have phytotoxicity conditions. And that's something we all want to stay away from. Like I alluded to earlier with bacteria, we don't have a lot of tools, so rotating materials is really important. There's been studies where we've got bacteria that are resistant to copper, which copper works on a ton of different components of a bacterial cell, so rotation is really important. I threw up this NUA fire blight model. This is it's called cougar blight. This is designed for the fruit tree industry. I haven't spent a whole lot of time with it, but we are dealing with very similar situations. Granted, we're selling the plant, not the fruit, but they are both impacted by it. And kind of putting this out to the group of collectively, if we could look at this from the nursery side of things and see if this is a good fit or a good IPM tool for us. I just wanted to bring this up to the group. All right, so so when it comes to this IPM program, this is the first year that we're utilizing this webinar approach. Steve Rettke has done amazing work, and he's really a great resource for this information. And now we're looking to scale up into utilizing traps. So the floor of my office is covered with traps, and we would like to have some people setting up traps. So we're going to do this on a first come, first served basis. It's for commercial growers in New Jersey, but we've got sticky cards. We could do some good monitoring with them. Like Steve was alluding to earlier, our clear wing borers are important right now. I have dogwood borer lures, and then I have a more generic clear wing borer lure. We've got wing traps, and we also have delta traps. We also have, as we move in through the season, scale insects are going to be really important, and we've got various forms of tape, different colors, different types, and we're going to use those for scale crawler emergence. So if you're interested in utilizing some of these traps, we really look forward to the opportunity to work with the industry to figure out what tools are best for us here in New Jersey to constantly keep these updates going. It's a great time too. If you're seeing something on your nursery, please feel free to reach out. We want to know what's going on. It helps us better inform and better understand what topics we really should be covering in throughout these presentations. So thank you very much for attending. I do see we've got a couple questions here. Like Bill said in the beginning, our next session is May 13th. So that's 3 weeks from now. We will put up whether that's a plant and pest advisor or an email blast, we will put out some information about the pest occurrences that are happening during that time frame. Next week, our next iteration, we're really going to talk a lot about scales across the various industries and some of the other diseases that are going to pick up in various industries as that temperature begins to increase. So, yep, just a slide about our diagnostic lab, and then one more. And if you're not signed up to the Plant and Pest Advisory, we're constantly trying to put new information up there. And a lot of what we've been talking about are Growing to Green Day based. And this is a guide that we put together. So I believe this could end the recorded portion of the presentation. And there are some questions